our project 2. So our project 2 is about plant growth analysis in R programming. So this is a plant growth is a built-in dataset in R. You can check our previous videos on YouTube channel, how you can be able to look at all the datasets, to explore all built-in datasets in R. So here are the all built-in datasets in R. From our previous project, we looked at air quality analysis project. This was our first project. So this time round, we are looking at project 2, and project 2 is about plant growth. You can check plant growth here. So this can give you a nice foundation on what we are working on, so that you can be able to build your understanding around the dataset, because understanding the dataset gives you an easier way of knowing what to do with the data. So yeah, so you can check here plant growth. This is not arranged in alphabetic order, so you can just check by the name, and then you can see here plant growth. So if you want to know more about here, you just see the name of the dataset and the result from an experiment on the plant growth. But when you come to, let's say here, and then type question mark like help, this is like a shortcut for help, and then you paste the name of the dataset, R is capable of now giving you full information. You can see on my right hand side in the help window, here you can be able to see that this is a result from experiment to compare yield as measured by dried weight of plants obtained under control and 2 different treatment conditions. So we have 3 groups of our plants. So we have plants that were controlled; they were not involved in any treatment, and then we have another type of other plants were involved or were growth in treatment 1, and another set of plants were also involved in treatment 2. So that gives us a few information about the format of our dataset. Our data frame is of 30 cases, so we have 30 observations and 2 variables. The first variable is numeric, which is weight of the dried plants, and the second one is the grouping, or we call it factor variable, which now in the groups it contains the following levels: contains the control, the treatment 1, and the treatment 2. And the source of the dataset is quoted here from the Dobson AJ 1983, an introduction to statistical modeling from London, Chapman and Hall. So once we now know the dataset we want to use, of course, now we have to start by calling this dataset. So at least by the end of this particular project, you'll be able to learn again how to load built-in datasets in R. You'll also learn to explore and summarize data, create some visualizations, and also build some compare groups statistically, perform one way ANOVA, and also you'll be able to interpret the p-values that we get from this data, and to actually be able to draw some data-driven conclusions from this particular project. So the objective of this research, the researchers wanted to determine whether different treatments affect the plant growth. That is exactly why we are here. We are here to determine whether different treatment affects plant growth. Now we'll start by loading the data. To load the data, you just call data method, and then you call the name of the dataset inside the quotes. So we'll call it plant growth. And we'll run this. You'll see in the environment right now is empty, but when I execute here, we are able to see that it has been loaded, and we can be able to see at least the first few rows of this particular dataset using a method called head, and we can now call the name of the dataset again. Okay, so when I run this, now we are able to see here in the console the first few rows in our dataset. So we said we have, this is numeric, which contains a decimal point, and then this one is factor. We'll call it categorical variable. Okay, so each row represents a plant and its recorded weight. Of course, the treatment it underwent, and also the recorded weight. Now we need to understand the structure of the data set. The way we did in the project one, we wanted to understand the structure. We want to understand the structure of the data set, which is very, very important to understand. The first step here is to, this step one is to load the data set to R. This is step two, which is now about understanding the data set. So to understand the data set, we have a few methods that we can put in place. We have str, and we call the object. So the object is the data set name, plant growth. Okay, so we'll execute this. You see some output here telling you this is a data frame, 30 observations, 2 variables. We had already seen that even in the explanation here. And also it gives you first at least some preview of your data, how your data looks like, and you can see how many groups do you have, or we call them factors. How many do you have? Like there are 3: control, treatment 1, and treatment 2. And we also have some other methods like dimension to check just the how is your data organized. So in terms of rows and columns, so we find out that our data has 30 rows and 2 columns. The next thing we do is to perform maybe some summary statistics. We call a method called summary, and then we call down the dataset to be able to give us the summary of the SEM. So we'll just do like that. We execute, just check. Okay, just check the summary plant growth. Okay, let me just check if this is correctly spelled. Let me see. Unable to find an inherited method for summary. So let me check summary. Okay, small s. Some of these things, yes, case sensitive, so you need to ensure that. Okay, so we can now see some summary statistics about our data. The first variable, which is weight, contains the minimum value of 3.590, and the first quantile, which is usually the 25% of our data, is at 4.550. The median is 5.155, and the mean is 5.073. The third quantile, which is usually the 75 percentile, is 5.530, and the maximum value in this area is 6.310. And then the other side, we have group variable, which contains the same number of observations across the 3 different groups. We'll call them level of groups. Now that one gives us the next step, which is now to look at, to explore do some exploratory data analysis. So we'll do some exploratory, explore some treatment data. So how to do that? We saw also in the other data in the other project. So if you have a categorical variable, let me call this just 3. You know, step 3. So 3 is we want to explore the treatment group. So the treatment groups here to explore them is just to get to understand them. Suppose we were not able to get this summary. This is how you can just work with the treatment group by using a table. It's like tabulate. So it's going to give us a table of plant growth, but now we are going to call the specific variable that we are interested in, which is the group variable, because this is the treatment groups. These are the treatment groups. So we have 3 treatment groups. So we have the control, which has, of course, we have already said the number of observations. So this actually shows that it's a balanced experiment, because each treatment contains the same number of plants. And so now that gives us a good step to start. Now, we might ask ourselves which group has the highest average to answer such like questions, and also which treatment appears most effective. So to answer such like questions, we will need to do some aggregation. We'll need to do some aggregation. For instance, that will lead us to something we call descriptive statistics, and we'll use now that question approach to now answer some of these questions that might emerge. So descriptive statistics help us answer the following. Descriptive statistics help us answer the following question. So it helps us answer the question, like which group has the highest average weight? Okay, let's take that as the question. So which group has the highest yes, which group has the highest average weight? Okay, so if you want to answer such like question, you need to use method like aggregate. So we need to aggregate. So we need first to, of course, we are aggregating by grouping weight by a variable called group. Okay, and then once we group using the variable called group, we are able now to refer, of course, to the data that we are using, which is plant growth. And then we call the method we are using, the method here that is going to be used in the aggregate, it's an aggregate argument, is mean. So we're going to use mean to perform this grouping so that you can be able to answer this question of which group has the highest average. So when we run that, the average weight, the highest group here is treatment 2. And you might be asked which treatment appears most effective. Now, which treatment appears most effective? So if these plants were dried and we wait, then it means we are interested in the weight. So the highest, the group that has the highest average weight is the one that is the most effective. So in this case, we'll say is answer is treatment 2. And when we look here at aggregate, so if we wanted to do some aggregation here, and say we wanted to know which group had the highest, so we were also able to see that the answer here is also treatment 2. Okay, so that forms a good foundation for us to get started with this particular analysis. So if you wanted also to do things like the median, the way we did in the first project to obtain things like median, it becomes now simple. Just use the aggregate. Remember, at least here you have answered some serious business questions or agricultural questions. But if you wanted to do some median, you can just remove mean and do the median. Just like this. If you want to do some standard deviation, you just change here to ST. That one can also give you those. So you are grouping this by their median, and you can see this one has the highest median. Okay, 5.435. Let's look at how they spread, which is now the spread. Do you remember mean and median are measures of central tendency? Okay, but now the standard deviation, SD, measures the spread. So let's check which plants has the highest spread, and maybe you can try compare some of the values we get with the mean and how does that affect the mean in reality. So when you look at the standard deviations that we have obtained from control is 0.58, treatment 1 has 0.79. So there's much variation on how far away are the observations from the mean. That's why they pull down something that relates to the central tendency. When you look at the treatment 2, you get that there is a standard deviation of 0.44, which is actually the lowest in this. So meaning that there is no much variation, there's no far away observations from the center. So that way is how we do use such like standard deviation to help us in such like analytics. In the next part of this project, we'll be able to look at now how we can do some data visualization, and then we'll also move from the data visualization to things like now hypothesis testing, performing ANOVA in the other parts of this particular playlist. So thank you so much for listening, and hope you can also take a minute to now explore your data in depth.